Well, I can see it, so
Yeah.
hopefully everyone else can.
Okay.
Brilliant. Should I just get started,
All right.
Zoe?
Yes, I'm going to mute myself, Inez, just to try and improve the sound.
Okay, brilliant. So firstly, thank you very much for inviting me to speak at this forum, which is very exciting. My name is Inez Januszczak. I am the sampling coordinator for the Darwin Tree of Life project based at the Natural History Museum. So my talk will be primarily talking about the progress when it comes to samples we've received or we've collected through the museum. However, I'll be doing a kind of a general overview as well. Okay, excellent. Next slide, please. Sorry. Thank you. So just in case you didn't know, the Darwin Tree of Life project is a really ambitious scheme to try and sequence the genomes of every eukaryotic species in the UK. So in total, this is round about 70,000 species, and we are currently in the pilot scheme. So there's a 2-year pilot scheme to try and collect various family representatives of these species. The aim is, once they're collected, is to sequence their whole genome. And of course, we're not doing this on our own. There's numerous partners. So I've mentioned some of them here, which mainly include the Wellcome Sanger Institute, which is the institute doing the whole genome sequencing, and as well as collectors based through the University of Oxford, Kew Gardens, and the Marine Biological Association. So the Natural History Museum started their involvement in March 2020, which, if you remember, is the start of lockdown. So unfortunately, the first year of the project probably went off from a bit of a shaky start, but things have picked up in the last few months. And I also just want to mention, I joined the project in June this year. So I won't be speaking so much about what happened last year, but I can give an update as to how kind of fieldwork and collection has progressed in the last few months. Just to recap, so the main difficulties with the project is that in order to preserve the whole genomes, specimens have to be frozen and processed on dry ice, which is at -80 degrees, to maintain DNA quality. As some people may know, we have received samples in ethanol. However, these won't be for whole genome, but for barcoding, DNA barcoding, which is when a short sequence of DNA is produced and is more used as an aid for identification, which I'll go into later. But I'm just making this distinction now, since I know people have sent in various types of samples before. Next slide. Thanks, Dave. So at the Natural History Museum, we collect specimens in various different ways. Sorry, Erica, I stole these photos from your Twitter, but these are from the Diptera Forum, which was, of course, a big event that occurred in June in Cornwall, where I think many people here would have gone and assisted in collection. So we managed to collect almost 400 specimens. So not all Diptera, there was a various mix as well. So those specimens would have been processed and also for barcoding. And within those, we managed to send a shipment at the bottom, which includes up to almost 500 tubes. Within those, 51 Diptera specimens from 23 families. So we do fieldwork collection. However, we also receive lots of live submissions and ethanol submissions from various collectors. Next slide, please, Zoe. Just thought I'd mention this here. We do receive lots of funny things at the museum. This one made me laugh because I received 10 tubes of carefully preserved flies, but with no name, no IDs, and no collector information. Just a very gentle reminder to people, if they want to send things to the museum, do include your names so I know who to contact in order to give you the proper credit when we're doing the processing. So we do accept specimens in ethanol, again, if live is not possible, because that is usually much more difficult for obvious reasons. And I found that Diptera don't tend to do very well in the post, which is something I can also go into later if anyone has any questions as to how they can send live specimens to the museum itself. Next slide. So like I mentioned, we're not the only group working on arthropods. White and Woods, which is based through Oxford University, they've also been collecting for a lot longer than we have, as they were fortunate enough to start their collection in late 2019. Liam Crawley has given me a small update on their progress, which includes 275 species from 45 families, including these few notable ones, which I won't try and pronounce because I don't know how to say them. But obviously they've had lots and lots of progress when it comes to submissions to the Wellcome Sanger Institute, mainly through White and Woods, but I believe they're also expanding their collection. For future involvement, we are also talking to collaborators in the University of Edinburgh, as they have a Sanger have sent also staff there to be based in Scotland in order to do more sampling in Scotland as well. So that's quite exciting. We have our collaborators are increasing, and therefore there will be more collection going across the UK, not just based in basically South England. Next slide. So in terms of the Natural History Museum, as I mentioned, it's been very difficult to sample in the first half of the pilot scheme. So we do have some data. Currently in our records, we have nearly 8,000 UK Diptera species recorded. Of these, we have 111 families. Currently, in terms of the Natural History Museum update, we are desperately trying to make sure our databases are updated more regularly, as we have been working with a bit of an out-of-date system where the families we have listed and the specimens we have collected have not matched. So this is taken from the live list, which I believe was circulated earlier on in the project. This is currently being updated right now, and from next week we'll have a much more accurate kind of description of what we have collected out of the sort of 850 family representatives that is the target. So at the Natural History Museum, we have 750 specimens of 317 species, which is quite good. However, obviously, if we want to hit that 850 target, we do have a little bit more to go. It's worth noting the reason why we have so many more specimens compared to species is we do encourage multiple collections, so of each species. It's quite common that people will send us 5 or 6 specimens per species, which is the right thing to do because we have found that not all the specimens are ideal for whole genome sequencing. However, this is only found out quite later on in the process. So hence we do encourage multiple species collection even at this stage of the project. Next slide. Thanks, Eddie. No. So this database that we're updating, I'm planning to circulate in future. Did this slide move? Oh, there we go. Excellent. Thank you, Zoe. So the reason why we are doing all of this, and this is the end game. So I mentioned that we want to produce whole genomes from frozen specimens. Now, White and Woods, I know, are ahead of us when it comes to genome notes, but I can proudly say that NHM had our first genome note, which was in fact a fly. So the St. Mark's fly, Vivio Marci, which was collected by a member of NHM staff. So it is quite exciting that this is the end result. So the genome note is essentially a published document that states the various bases of that particular species. So this is what they look like, and this is what we're aiming to have, 850 versions of. I will say that this fly was collected last year, so it has taken, almost a year to get to this stage. There will be some time before we get all the genome notes of the submissions, but it's very, very exciting that we are making some progress there as well. Next slide. Thank you. So I thought I would summarize by saying, for more information on how you want to get involved, or if you want to submit any specimens, do email the Darwin Tree of Life email address. This comes to me and our entire sampling team at the Natural History Museum. And this includes if anyone needs any equipment, so things like tubes or tubes and ethanol, we're more than happy to provide those. In terms of what has been collected and sequenced so far in the project, like I've mentioned, our Natural History Museum database has been a little bit out of date, and we're currently trying to update this. However, if you want to see what has been sent to the Sanger Institute, this is available through the Darwin Tree of Life portal, which contains all of the information in terms of the samples that have been sent there and that the samples that are in the freezers, as well as updates on how far they are in the whole genome pipeline. As mentioned, once updated, I'm planning to circulate this live list. We're also planning to include specimen preparation data. So how many samples we have in ethanol, how many samples we have in the freezer itself. And once this is done, I think we'll have a much better representation of what we have and what we need. The species list collation has been a challenge, and we are trying to work more with White & Woods and with other arthropod collectors to coordinate better what we are collecting. Since up till now, I think there's been a slight, it's been a bit difficult to track what people had in the pilot stage of the project. But moving forward, we're definitely planning to do more targeted sampling. So if anyone would like to have that information once it's available, I'm more than happy to provide it to Zoe to circulate as well. But feel free to email me and the team for any questions that you have if you do want to get involved in the project. And of course, for everybody who has submitted something, thank you very, very much for your support, and I look forward to continuing to work with you in the future as well. And I thought I would include my favourite fly species, which is of course the scorpion fly, as an ending note. Thank you very much. Oh, can't hear you, Zoe. So, well, I discussed with Zoe about the possibility of doing a kind of a question answer at the end.
I'm not sure where that's in trying. So sorry about that, Inez.
That's all right.
Yep, that's the intention. So let me. We've got a couple of questions. So the first one from Darren Williams is asking where we're doing the sequencing.
So they're sequencing at the welcome.
So that's could you say that again? Could you say that again, Inez? You just broke up.
The whole genome sequencing is being done at the Wellcome Sanger Institute. So that's where all our specimens will be sent after they've been processed. So we don't do any of the whole genome sequencing at the Natural History Museum. However, we do the initial stage of the analysis, which is the DNA barcoding, where a much shorter sequence is produced.
Okay, so we're doing a small amount at the museum, but the majority is being done at the Sanger. Okay, and there's a question here from Gabriel Nuvi. It says, what is the final aim of the Tree of Life? A complete phylogeny? And are there any unexpected results to date?
That's a really good question. So I mentioned the whole genome. Obviously, getting the genome note is ultimately the short-term goal. However, the ultimate goal would be if we are able to monitor the biodiversity of all of the species in the UK, that would be a huge advantage and an incredible record to have, especially since measuring biodiversity is something that is currently being prioritized as a measure of the health of the environment. So I think long term, if we have the genomes of all of the species, this has huge implications for how we monitor biodiversity, and how we do future biological sampling, especially as there is so much progress in things like eDNA, using smaller bits of tissue to see if we can identify species. So I think there's the identifying species aspect of it. There is the monitoring species aspect of it. And like you mentioned, there have been some unexpected results in the sense of species which were, unfortunately, I can't think of any examples off the top of my head, but there have been cases where species that were identified as one. Oh no, I do remember in annelids. So there's an Insignia ardeni. There's a type of worm. So the striped and non-striped, which people thought were two different species. However, they've never been sequenced before. So this was a morphological assumption as opposed to a genetic one. And I think the more genomes we sequence, we will start to get more and more examples of perhaps species which were put in the same family or in the same genus that may turn out to not to be the case, but simply because no one had actually studied their genomes before.
Oh, that's interesting, yeah. Okay, so we've got some more questions coming in. One from Mike Ashworth. How many of the barcoding specimens have been done, and will they end up in bold?
That's a very good question. Thank you, Mike, for your question. And Mike submitted many things to the project, so thank you for that as well. So I can't, off the top of my head, tell you how many we have barcoded. What I can tell you is each specimen has about a 70% success rate. So by that, I mean passing barcoding, then having a merged sequence, and then having a match on bold. So it's a three-stage process. Once the DNA barcoding, if it is successful, then it will go on bold, yes. However, the sequence has to be of a high enough quality to be accepted by bold. And that is something that is very variable and depends on the species or depends on the preservation. But yes, anything that is submitted from DNA barcoding, the aim is that it will be put on the bold, which is an online database, for those who don't know, of genetic sequences, which means people all around the world could use it to identify species as well.
Okay, so am I right in thinking that bold have set up a whole bunch of criteria now that to try and make sure to curate what gets onto the database to stop any sequences that aren't particularly good ending up in the database? Is that right? There's now quite a lot of.
So I would say that's half right, Zoe. So bold do have a criteria. However, that criteria isn't particularly strict. So the Natural History Museum, we will only submit sequences to bold that are merged, which means that when the DNA is being amplified and then put through, when the DNA is being amplified, it has to be a certain state in order for us to deem it acceptable to go on bold. And this is generally the case. However, what we can't control is if people put misidentified species on bold, because you have situations where the sequence is good, however, the identity isn't. But that is incredibly difficult to monitor because bold will accept it if the sequence is good. There are cases when you will have a very, very strong identification and a strong sequence, but it will match to an assortment of species. And this will require manual cleanup. So we are currently talking about whether we should be telling bold if there are clear errors in their database. But this is not something that bold can do automatically. It would require people looking at the database and pointing out these errors.
Okay, that's interesting. Right, we've got another question from Darren Williams. If we have this information, so I think he's referring to the genomes and the sequence. If we have this information, we understand basic biology, i.e. physiology and development. I think that would require us knowing what all the genes in the genome are doing, isn't it?
Sorry, what's the question? Sorry.
It says, if we have this information, we understand. Here we go. A statement, not a question. Sorry.
Like, how do I answer? How do I answer that? Yeah.
I think we can compile these genomes, but we don't necessarily know what all of the genes in the genome are doing, do we?
What does he mean by rules of the genome, I suppose?
Well, no, it's just it is just a statement. He said, if we have this information, we understand basic biology, i.e., physiology/development. But I'm not sure that we do we? Because we don't know what all of the genes are doing, do we?
So again, the end goal is, yes, if you sequence everything, you will have a much better understanding of how species relate and how their genomes relate. But we're nowhere near there yet. And I think, like you mentioned earlier, lots and lots of things will probably come up the more sequences we generate. And what's also true is currently there's limitations. There's limitations in size, there's limitations in preservation. But the aim is, once we have done the collecting, hopefully the technology will improve enough that we can sequence, for example, incredibly small specimens, or we can sequence specimens that have not been preserved in -80. But that's a few years away. So yes, there's definitely a lot more work to be done.
Okay, we've got a comment from Liam Crowley here. He says,
Hi,
Liam. Darwin is referring to the difference between annotated genomes and unannotated genomes. And a long question from John and Barbara Ismay. Do we check identifications with experts before you submit? We have seen wrongly identified chlorophyds in pretty much all museum collections. Also, we know that 2 different species, non-British, share the same barcode. We think this is because chlorophyds have evolved recently and rapidly. And it said, okay, so this is from Barbara, not from John Ismay, so from Barbara Ismay. So are there cases where barcodes don't work for species identification, do you know?
So again, our policy is if the sequence is good, we will submit it to bold. The ID that we use then depends on who submitted it and whether there are any matches. If there are no matches on bold because there have been no sequences submitted before, we will use the ID given by the expert identifier. And for us, expert identifiers mean trusted members of the community, trusted curators at the museum, people deemed experts in their field. So we will not put an ID through that has not been validated by various people. So if things have been misidentified in the past in bold, this is exactly the kind of thing which we want to combat. Hence, again, it's important we have multiple specimens, all verified by someone who is an expert in that field, so we can improve the bold database.
So the end result, will it be that people looking at the data that is generated by DTOL will be able to see the person who made the identification? So that will happen?
Yes. Well, not only that will happen, that is happening. So there's been cases where we have had expert identifiers identify something and it has matched to something different on bold. However, they then checked who did the bold submission and found it to be another person in the field who works in a different institution, for example. And in those cases, it's very much, there needs to be a conversation, and there is an element of trust there as well. So yes, anyone who submits something to bold, the ID, their name will be attached to the record, so people will be able to see who submitted it.
Okay, I think we're going to have to make this the last one because it is 11 o'clock. We've got a clarification from Liam Crowley, and he says, barcodes are also assessed in a phylogenetic framework, i.e. draw a gene tree to ensure the ID is accurate.
So I would say to Liam, this is true when you have enough genomes. However, there's been cases where we put a tree together and there hasn't been enough sequences on bold to fill out the tree. So I think the tree is good if you have enough of the data, but this isn't always the case, as mentioned earlier as well. So I think it's a good tool to have, but I wouldn't use it as the only tool.
Okay, right. Thank you very much, Inez. That's great. And we do need to move on now because it's 1 minute past 11 and it's time for Roger Norris. So
Great. Thank you very much.
Thank you very much and goodbye, Inez. Bye.